Hello everyone, my name is Rahul. In the previous video, we had discussed about gene therapy. In this video, we are going to discuss about molecular diagnosis. Molecular diagnosis means identifying, identifying or diagnosing the disease with the help of recombinant DNA technology or genetic engineering. So normally what happens is we diagnose a disease only when there are certain signs and symptoms present for that particular disease. So if a virus or a bacteria or a fungus, whatever organism enters the body, it will have a phase of pathogenesis when it will multiply inside the body. Then it will produce certain signs and symptoms when its concentration or when its population will cross a certain number. Now, there are certain diseases where early detection of a disease will help to change the course of the disease and provide very good benefits to the patient. So, for an effective treatment of a disease, early diagnosis and understanding its pathology and pathophysiology is very important. Using the older methods, using older methods such as urine analysis, serum analysis, etc., we get results very late. By the time we get results or by the time we are able to detect the presence of microorganisms using these older methods of urine analysis, serum analysis, etc., early detection is not possible because the population of bacteria or virus has reached a very high amount, which is why it is detectable in the serum or the urine or feces or whatever we are using. So using recombinant DNA technology, we can help in the early diagnosis of a disease. So in this, the first method in molecular diagnosis is PCR. PCR, as we already know, it stands for polymerase chain reaction. During the COVID times, you might have heard of RT-PCR report. Everywhere it was necessary an RT-PCR report. So PCR stands for polymerase chain reaction. We have already discussed about polymerase chain reaction in detail in the section on tools and instruments of biotechnology. So in PCR, what we do is we amplify the genetic material of microorganisms. This means that, therefore, even at very low numbers or population, the presence of microorganism can be detected. We already know that in approximately 30 cycles, the genetic material is amplified 1 billion times. So if a bacteria or virus is present in very low population and very low numbers, we can just take its DNA or the RNA, whatever it is, its genetic material is, and we can use the PCR technique to amplify it, and we will be able to detect this pathogen at very low population. PCR, it is now routinely used to diagnose HIV. It was widely used to diagnose COVID-19. It is used to detect mutations in cancer patients. these are the uses of PCR. It is a powerful technique to identify other genetic disorders, because we only need very small amount of DNA, and it can also be used in forensic studies. For example, from a crime scene, we obtain only a single strand of hair. From that single strand of hair, we can amplify its DNA multiple times so that we can experiment, or we can identify the culprit or the victim, whatever we need, because we have amplified the DNA. So a very small sample will suffice in our investigation. Next is DNA probes. What we do in DNA probes is a single stranded RNA or DNA, whatever we are trying to identify, a single stranded RNA or DNA which is tagged with a radioactive molecule, which is known as a probe, is allowed to hybridize with its complementary DNA, followed by detection using autoradiography. Now let us take an example to see how this works. For example, we have this gene, and this is the mutated gene. So this is the normal gene and this is the mutated gene. We identified a disease where this will be the mutated gene and this will be the normal gene. Now we have to see whether a patient contains a normal gene or a mutated gene. So what we will do is we will take a single stranded DNA or RNA which will be complementary to this mutated gene, and we will add a radioactive dye or radioactive probe to it. Now, under conditions of heat, what we do is we will denature the patient's genetic material. So patient's genetic material will now be converted into single-stranded DNA. Patient genetic material will now be converted into single-stranded DNA. Now what we will do is we will introduce this probe to the patient's body, not body, to the machine or to the instrument where we are performing this diagnosis. We will introduce this probe. Now this probe will hybridize with its complementary DNA. Now if complementary DNA is present, it will hybridize. Here it will not be able to hybridize because complementary DNA to the probe is not available. Now we will detect, or we will scan using autoradiography. We will have an instrument which will be able to detect this radioactivity, and if this radioactivity is seen in the patient's DNA, we will know that the patient's DNA contains a mutated gene and the particular patient is suffering from that particular disease or a particular type of cancer. In this manner, we can diagnose the mutation, we can diagnose the cancer, we can diagnose a mutated gene or a genetic disease. So the clone having mutated gene, clone having mutated gene, it will appear on the radiographic film. It will appear on radiographic film. In this case, we have taken a probe which was complementary to the mutated gene. If we take a probe that will be complementary to this normal gene, so there will be no film on the radiographic film. It will not be able to detect on the normal radiographic film because we are now taking the normal gene. Next is ELISA. ELISA stands for enzyme linked immunosorbent assay. Enzyme linked immunosorbent assay. ELISA can be of two types. It can be used to detect antigen, or it can be used to detect antibody. Antigen refers to any molecule, any substance that will be present on the cell, and in response to antigen, antibodies are developed. So we can use ELISA to detect either the antigen or the antibody. So infection by pathogen, it can be detected by the presence of either antigens, which can be a protein, it can be a polysaccharide, it can be a lipid antigen, it could be any antigen, it could be any molecule, or we can use it to detect the antibody. How this is done is, what we will do is, we will take, for example, we are trying to detect the antigen. So what we will do is, we will take a slide which has been coated with an antibody against this particular antigen. This slide has been coated with an antibody against the particular antigen. Now we will add the sample of the patient. If the sample contains the antigen we are trying to detect, that antigen will come and bind to this antibody. Now after this binding, we will add a substrate that produces color or changes color in the presence of an enzyme. If this particular antigen antibody reaction has taken place in the presence of the substrate and enzyme, there will be a color change, and the intensity of color change, it will give us the quantity of antigen present in the sample. So this ELISA, it can be used to both answer the question whether the disease in question is present or not, and it can also be used to accurately quantify the amount of antigen present in the sample. So with this, we finish off with this discussion on molecular diagnosis. See you in next video.